Good morning. My name is Abby Bell. I'm the farm manager at Alemany Farm. I want to welcome you all today to the Friends of Alemany Farm's workshop, Seed Sowing and Propagation Magic. We are very, very excited to be partnering with Edgar Xochitl today, who is the current farm manager at Hummingbird Farm and who has a wealth of experience and knowledge about seeds and plants to share with you guys today. We're very lucky to have this experience. If you have not read Xochitl's bio in the workshop description, I strongly encourage you to do so. And please check out Hummingbird Farm, which is a project of Poder. Before we begin today, I just want to express deep thanks for everyone for joining us and for your financial contributions. We were so encouraged by the offerings that were made in our first virtual workshop a few weeks ago. And it's because there was such high interest that we're able to continue offering these virtual education workshops today. So thank you for all those that have been able to contribute. Today's workshop will be divided into 4 sections. Each will be a topic to help you sow seeds and propagate plants. Xochitl will show a video demonstration for some of the topics, and after each section there will be time to answer 1 to 2 questions, and then at the end of the workshop there will be more time for questions and answers then. Please use the Q&A box to ask your questions. We have over 100 people attending today, so we may not have time to answer all of your questions, but we hope to get through as many as we can live or written, and if needed, some after the workshop. This workshop will be recorded, and we will also send out the recording with a follow-up email and a survey, an anonymous survey, and any other necessary resources. Please keep in mind this is only our 2nd virtual workshop, so bear with us if we experience any technical difficulties. Without further ado, I will turn it over to Xochitl. Thank you so much.
Good morning, everyone. My name is Xochitl, and I'm here to share some of my propagation information I picked up along my experience doing this. But yeah, I have a little presentation for y'all. I'll wait till that gets pulled up, but yeah. Again, thank you everyone for showing up. We're a new paradigm shift, so this is part of how we grow and cope with what's going on. So, and plants help for that. Plants are a great way to ground yourself figuratively and literally. So, yeah, I'm gonna wait for the next. There we are. All right, propagation magic or propagation at home. So, go to the next slide. Okay, so before I get started, there's a description of me on the bio to the workshop. I just wanted to add a few things. In 2016, I was a propagation specialist at UC Santa Cruz for the Center for Agroecology and Sustainable Food Systems. That's actually where I met Abby. So, and then one of the highlights I did there as a propagation specialist, I led the plant sale, which is the largest fundraiser organized by the farm on campus. And the theme that I organized around was decolonizing flowers. And that was looking at the legacy of colonialism and how we label and name our plants, both scientifically and culturally. And I currently now I'm in San Francisco doing some edible landscaping around the bay, as well as the farm manager at Hummingbird Farm in the Excelsior of San Francisco. Next slide, please. So like Abby mentioned, there'll be 4 sections to this workshop. First part is seeds, what they are, their mechanisms of what's going on inside, materials to propagate, so various examples of materials you can have around your home or around your growing spaces of what will help with your propagation at home. as well as just germination methods, two that I use frequently. Broadcast sowing is a video, and then I'll just give an example live of transplanting in individual cells. And then just some advice on planning for time and space logistics, especially as you're doing this at home, how to navigate the space around your home so that you can maximize your greenhouse potential. Next one. Next. So what are seeds? And this is there more to this? Oh yeah, it's just an image. All right, so seeds are, you know, they come in different range and sizes. And so the first part, before we even consider propagation, we should really look at what seeds are. So next slide. So seeds are the offspring of flowers. And I think this is super important to acknowledge. Because in these offspring, there's a few things that come out of these seeds. One is the genetic memory of these plants or flowers, and that includes their relationships with other organisms. So in the seeds, plants store how to interact with other organisms, whether it's for defense, attraction, or disbursement of seeds. Examples of defense would be creating thorns, physical alterations, or chemical alterations to defend themselves. Same thing with attraction, figuring out how to manipulate their own, or how to use their resources to attract it, and then later on disperse their seeds. And within that, there are also the adaptation. So like I mentioned briefly, biochemistry. So that's what they're using to physically among their photosynthesis, how they're channeling that photosynthesis. Also the adaptations to competition, resiliency, as we look at competition and resiliency among themselves and other plants, but also just adaptations to the climate. And this is something that we get at a sexual propagation, other than, in this case, seeds. Something at an asexual propagation is that you don't actually get new adaptations, you just get clones of that plant. And I'll talk a little bit more of that later. Also within seeds are the food reserves of these plants. And this is only part of most plants where they're heterotrophic. And what I mean by that is that the plant or this baby seed has to eat. It has to consume energy similar to us, as well as fungi. We are heterotrophic organisms, which again means we have to consume resources to grow. And at this point in the plant's life, the baby in this case is heterotrophic, and that's essentially what the flower and the previous generation is doing, is providing the resources and nutrients so that these seeds can last dormancy, as well as have enough resources to germinate and reach the surface. And the food reserves are essentially the carbohydrates or sequestered carbon from that the plant has enlaced with enzymes to get that process going. It's also a source of nutrition for various organisms, birds, bugs, mammals, humans. We use seeds for various sources of nutrition. And they are harnessed plant magic. And what I mean by that is that the first seeds in gymnosperms, which are simple pine cones and whatnot, came across 380 million years ago. Angiosperms, what we know of as most of our flowers, came across 125 million years ago. And just to give a little perspective within that, the grasses, which is the bulk of our grains or seeds that has allowed humans to expand in an agricultural context, showed up on Earth 10 million years ago. And then humans showed up 7 million years ago. And then even within that, we have a fairly short amount of time as humans interacting with seeds as a permanent relationship to our structure. So, and then another thing I want to add to this is that with just similar on this topic, but a little off, is that there's no seeds located in fossil fuels. So all the fossil fuels that that we deal with in our current society are results of spores, of plants that came from spores. So ferns and early fern trees, maybe a little bit of early cycads that were producing early seeds here. But most of the seeds that we that exist on Earth today are fairly recent additions to our planet. Next slide, please. So seeds come in different sizes, and they also have different lifespans. So all those carbohydrates and enzymes have a shelf life, and this is just an example of plants that you will most likely be germinating at home. A lot of these are edibles. Well, they're all edible actually. But as you can see, not all seeds are created with longevity in mind. Some of these are a much quicker life cycle, so they need to begin their next life cycle fairly quickly. And these numbers are just rough estimates. They may be some seeds may, depending on how they're stored, may have longer shelf lives. But for an average, most seeds that you probably have around your house could be good for about 4 years, I would say 3 to 4 years. The first two, corn, for example, in this case, it has a 2-year life cycle, but that would be more if you're having it, if you're taking the individual kernels off the cob, you'll have a longer, you can extend the shelf of your corn by leaving the corn on the cob. So yeah, so this is just a general info slide. Next slide. Here's some terminology that just comes in handy whether you're reading a label for your seed packet at home. And I think these are the 5 top terminology I feel everyone should have a general understanding of. The first one is viability, and this is the plant's ability to germinate. And again, this is a result of the resources that are encased in those seeds. Dormancy. Some seeds need dormancy to germinate. And this is part of their evolutionary process where they understand and they've evolved to undergo a time of dormancy because the climate is suitable to grow. So this would be like in northern latitudes, that would be a winter or a cold period, and in warmer, more hot areas, this would be times of drought or just extreme heat. So it's an advantage to become dormant to ensure your survival. Stratification is some sort of treatment to break dormancy, and that could be a combination of things. It could be acidic, so that would be seeds going through an animal's digestive system, or they could be some sort of reaction to a forest fire. So the ashes and all the chemistry that's left behind after a fire mixes with water, and then it enters the soil and it stimulates seeds to break their dormancy. Another stratification procedure is physical, and that could be just physically breaking seeds in one way or another, the outer shell. And this allows water to seep into the actual embryo and then facilitate the activation of enzymes and whatnot. And the last one is temperature. So this would be, from going from cold to hot or too cold for warm, and this would mimic the transition from winter to spring. Or there's also just going from hot to cold. So you would, for plants that would germinate in the fall, over winter, and then continue growing in the spring. And then there's ways to mimic that. You can put your certain seeds, and a lot of these instructions would be in your seed packets. So not all seeds need a temperature change, but for those that do, you can put them in the fridge for a while and then take them out. And that could be both leaving the packet in the fridge or sewing a container and then putting that in the fridge and putting it out. So soaking softens seeds, coats, and increases viability. So this is just adding water to your seed, and this is really helpful with larger seeds. So beans, peas, cucumbers, different squashes, and whatnot. And what this does, it allows the water to enter the seed. It begins just by osmosis. Water enters slowly, it expands, and it just increases the enzymes that are already present in our plants. And then some seeds will need light, and this is more like I'm thinking chamomile, feverfew, purslane. A lot of these seeds, the character of the parent plant just drops the seeds on the surface, and they require a combination of soil and light and water. But for the most part, I would say that temperature is much more needed than light for most seeds. Next slide. Germination does mean the sprouting of the seed. So here is just an example of what's happening in the seed. As you can see, the depth is about twice the size of the seed. And this is super important because as you can see in the different transitions of this plant, of this bean, for the bean leaf, the lighter green leaves on this image, those are the cotyledons. And what that means is those are the sources. That's where all the energy is in that seed. And so in this phase of the plant is in this heterotrophic, it's consuming energy, it's consuming all the energy out of there. And these seeds need to be able, those cotyledons need to get to the surface so that they can begin photosynthesis. And this is when the plant, and this is the transition of magic, I would say, between the plant going from having to consume energy to producing its own energy from the sun. This autotrophic approach. Next slide. So yeah, this goes into our next section. So let me, is there a question? So there was a question.
Sochi, there's a question that a couple people have asked about if all organic seeds are the same, or are there certain aspects we should look for when we are purchasing the highest quality seeds?
That's a very good question. So for highest quality seeds, you would want to go with the most recent plant generation that you could get. That'll just increase their viability. But as far as organic seeds and whatnot, to have the label of organic means you have to go through certain loops and ability to have the certification. The plants themselves will be stronger because they don't have any additional inputs chemically, so their own genetic memory is better suited for the long run. For certain crops, seeds may not be available organically. You might only be able to find conventionally. As far as seed sources, there's various different seed catalogs that kind of emphasize on organic or better ethical seeds. But I'm trying to think of I don't have a straight answer for that because it really it depends on where you're getting your seeds, and um, most seeds are found in packets. So it really, if it's that important to you, I would start by going for seed companies that fall along your own ethical needs. Um, I hope that answers that question. If you buy non-organic seeds and grow them organically, yeah, they are considered organic. So the input, again, the conventional aspect to them are really from the inputs that are being put into them. The GMO aspect to it is more because you can have something that could be GMO, and that's more of it's been sliced and diced at a genetic level versus something that could just be conventional and an open pollinated hybrid. That means that it's been pollinated in a way that is still crossing two plants of the same kind versus genetic material from a completely different organism. But if you grow them organically through one season, for the sake of just open pollination and resiliency, you've created a new generation. And you could call it organic, it just won't have a label, an official label of organic. But yeah, so next section, and I'll have more time for questions and answers later. But germination methods, there's two, actually, let me go back. There's two main ways to go about this, broadcasting, sowing, and then individual trays. Broadcast sowing, essentially, you just take your container and you sprinkle your seeds on the surface, and I'll have a video of that showing that shortly. But the pros behind this is that you get a lot of seeds real quick, and for seeds, may have a low viability, I prefer this method, so I can just use the entire packet, and then since there's already a lower germination rate, by increasing the amounts of seeds, I might even out with what I'm trying to go for. I also have a lot of seeds that are like 2014 older, so I just tend to do all this in broadcast. Just again, trying to maximize my viability with my time and resources. It uses less space to begin with, and it provides extra competition because all these seeds are together, so they kind of speed up a little bit in trying to fight to obtain the most nutrients. The cons to this is there is no social distancing, and what I mean by that is that if one of them gets sick, because there's nothing physically breaking them apart, they'll all get sick. You can have a large amount of numbers to begin with of plants, but then you can lose them all pretty quickly at the same time. And then you will need to invest the time later to prick them out or to move them out of this confined space into something much more satisfying for each individual plant. Individual trays, each plant has its own cell. It helps prevent the disease. One of the big cons to this is it just takes up more space at the very beginning. And so if you're limited on space, I find it easy to do broadcast sowing, a lot of different broadcast sowing, and then prick out your trays of what you'll need. But to each their own. And again, it depends on the materials you got around. So next slide. Water needs. The main advice I would say is that just keep your soil as moist as a damp sponge. You want to keep it as consistent. So that again is going to vary on where your plants are at and how much evaporation is happening from baby seed or the soil itself. So when the water hydrates the enzymes in the seeds, activating them, and then this is essentially what starts germination. And through osmosis, the pressure builds, and then cells enlarge. And then overwatering can cause your seeds to rot. If there's an excess of bugs around your soil medium, you probably are overwatering. And overwatering can cause your plants to rot. So, can we go to the next slide? I think it should be a video. Now this is just a close-up, but like I mentioned, the cotyledons must reach the surface to begin the process of photosynthesis. And this is super important because if the plant is sown too deep, it'll never reach the surface. It'll eat all its energy reserves and die. There's only two types of organisms that are autotrophic. It's photosynthetic organisms, so that's algae and plants, and then organisms at the very bottom of the ocean, chemosynthetic, which are like bacteria that live at the bottom of the Earth's bed. But everything else needs to consume energy, and then autotrophic organisms create the basis of all our food chain. So 2 times the depth is how far you want to go. So that, and again, depending on the size of the seed, smaller seeds will be higher to the surface than bigger seeds. Next slide. Here's their sowing materials. There's a various materials you can use. Some to buy. So you have peat pellets, miniature greenhouse, cell trays, biodegradable containers. You can also use plastic cups and just make holes on the bottom, or you can create your own miniature greenhouses out of this little takeout container and some milk jugs. So next slide. Here is an example of broadcast sowing. So into the left or the circle image, you have a quarter of each of these pots being dedicated to a different type of seed. And to the right in this box frame, you have just one variety of seeds taking up the entire section of the entire medium, or the pot, the flat in this case. Next slide. Oh, yeah. So here's an example of broadcast sowing. So those are the seeds I'm using. I know the angle's a little off, but I'm really just breaking these seeds apart and sprinkling them across the surface of the tray. Yeah, I guess the video is choppy. This is similar to the image I just showed earlier. Can we come back to the screen here? I'm going to talk a little bit about soil medium and material. So when you're looking at soils, I make my own germination mix, and it's a 3-2-1 mixture. It's 3 parts peat moss or coconut coir, 2 parts compost, and 1 part sand. But a general potting mix that you find should be good for home. I like adding sand into my germination methods because when I prick them out, it's easier for the plants to come apart. So we're working on trying to get feedback on this image. So soils. A store-bought mixture can work just as well. And each different material that you're putting into the soil medium is going to give you different properties. The peat moss, again, it's not a fully sustainable resource. They're taking it out of a bog, most likely in Canada. But it does give you some good water retention. So a good substitute would be coconut coir, but unless you're buying it on your own, you probably won't find it in a mixture of soil. But here, this is just soil here, right? Another material that's added into yours, perlite. Perlite allows for aeration, and this is things that are already going to be found in a conventional sowing mix. The only thing if I do buy a conventional mix, I just add a little bit of sand into it, usually about a third or whatever I'm going to use. And the sand helps with drainage, and again, it just allows the roots to grab onto the particles, and when I'm trying to separate them, it's easier to break them apart. And there was another thing with soil. Yeah, you want the peat moss or the coconut coir. Again, it helps retain that moisture, and I think that's what you want in your baby seeds. The peat moss, I just thinking of something. They also, if it's like I mentioned, if it's too much water, they're hanging on to the moisture where the insects are coming in and most likely laying their eggs in the peat moss. So this is if you can make your own compost, it'd be great. And another thing that, because the roots are starting in the soil first, this is where the plants need to figure out how to build their social skills and how to interact with other organisms. Because before plants can interact with their leaves and everything, they have to build the connections with the microorganisms in the soil. So another thing that I tend to add, and you can find different versions, but this is just a mycorrhizae. So I'm not even worried about the brand, but essentially it's just a powder. And essentially what's in here is a combination of different beneficial microbes. So it comes across as a powder, and you basically just need a little cap for about a gallon. It's 1 scoop for 2 gallons of water. And so it's just a tiny little scoop. I've had this for over a year. It's just 5 ounces, and I use it on my sowing. But I just find it useful so that it doesn't, so your plants have beneficial friends already waiting for them in the soil. And what's happening, because plants are able to photosynthesize, they're exchanging carbohydrates for minerals. And so they're giving minerals to the fungi and to the bacteria that are helping break down soil particles into the nutrients that plants need. And in return, plants give these fungi and bacteria sugars, which is, it's the early part of negotiating and building a community together. One thing I wanted to show so if you have, this would be a 6L that you could use. And again, this is a bigger one. You can find smaller ones. And here, you can see if you grow a seed individually, there's a physical barrier to block disease or anything going in between one to the other. Let's see. Do we have questions on this part? It's a lot to cover in a little bit of time. So plus I'm at home, so thank you for your patience. But the packet, it is just, this one's called the wild root. But again, it's a mycorrhizal fungi. That's more what you want. There's different brands. There's Root Shield. There's like Endo
There's
Micro.
a question about if potting soil is different than the soil you need to germinate seeds, or if you could just use a store-bought potting soil.
That's a good question. Potting soil is already designed to, it's got a different combination of materials already to facilitate the survival in a pot. Is it the same as a sowing mix or germination mix? For the most part, I would say yes. The materials are well uniform that you could start seeds, unless it was really like, unless your potting mix had a lot of larger wood chunky chips. As long as you could see a baby seed pushing itself. You don't want anything too heavy that's gonna inhibit the seed from being able to reach the surface. So again, a small little twig, although it may not seem like much, I mean, this probably is not that bad, but if it was bigger than this, I don't really see anything here. It might just physically block the little plant from reaching the surface. But I could see that. I could see how both of them could be used for germination. A potting soil could be used for a germination mix. Again, I tend to use sand. I just add sand and not beach sand. Let me emphasize that. You don't want to add salts to your soil. So it would be some sort of a horticultural sand or river sand. You don't want salts in this.
There's another question. Should plants be placed or seeds be placed under a grow light immediately after germination?
If you have access to a grow light, yes, that would allow their stems to have a better strength because they'll follow the light. If you're having, if you put your tray next to a window, I would just suggest you rotate the tray so that the plants continue to move towards the light in opposite directions. Does that make sense? So if I had, let's just say I had my window, this on the window, and then and every day or every other day, I would just flip it so that the plants strengthen their stems in the opposite direction. And at the very start of the process, consistent temperature will unless the seed variety needs light to germinate, and again that will be found on your seed packet. The consistent temperature is much more important.
There's one more question about when is it best to use a greenhouse? Can you use it for all seeds? When should you take them out of the greenhouse?
Yes, so a greenhouse is a structure that not all of us will have access to. But if you do have access to a greenhouse, this allows you to extend your sowing season. So if something, because essentially you're creating a bubble of temperature around your plants. So yeah, so depending on the variety of seeds, for example, the tomatoes that are being sown now or transplanted in the ground, most of those started in February. And again, it's too cold for most plants to germinate then, but inside a greenhouse, you can start some of that process earlier. Yeah, if you have access to a greenhouse, you can plant all kinds of materials out of their season. And again, this propagation section is mainly on crops that we'll be using for food, but if you were trying to grow other sorts of plants for other reasons, a greenhouse is a great resource. Also within that, I do want to mention that greenhouse technology is a result of colonialism. It's a result of having people explore and bring back plants from other regions of the world, bringing them to Europe, and then creating infrastructure and technology to allow these plants to live outside their ecosystems. They're used now in various ways to benefit us in an agricultural context, but that is part of the legacy of where that technology comes from. So I just want to mention that.
One last question in this section is there's a number of questions asking if you could repeat them, that you were adding mycorrhizal to the, if you could show that again and,
Yeah,
and
I'll
repeat.
type it in the chat box right now, but it's just mycorrhizal. This, again, there's different brands, so I'm not promoting this specific brand. But essentially what this is just microbes. It's, you know, according to this, it says 120 million parts per million of microbes in a little teaspoon, or a little scoop. And it's probably an ounce. But I will write it down on here. Michael. And if you just do a general Google search, you'll find something. Again, I don't remember how much this costs, but I've had it for over a year and I use it pretty consistently. So maybe it was like $50, but again, over a year. But I'm also germinating large amounts of seeds, so you might need something smaller, or find a few folks you can go in on.
Do you want to go to the next section and
Yeah,
come back to
let's go to-
questions at the end of the workshop?
Yes, yes. Can we go back to the PowerPoint? So here's some, again, just what happens with broadcast sowing. So you get a lot of seeds in a certain container, and you can put, you know, to the left here I have sunflowers, to the right I have tomatoes, and you know, in each of those there's easily 20 plants, and it only took a 4-inch pot. So the next part of that, once you already have your seeds germinated, again, if you had old seeds, this is a great way to just use up all your old seeds, and find a 4-inch container. If you know, just if you think they're the seeds that may not be viable, similar according to the chart I posted earlier, just try them anyway. Seeds have, you know, those are just general averages. You know, the beauty and survival of these seeds is that they're all different. They're all individuals, and so some of them are late bloomers, and that late blooming is a survival tactic. So give them a try. So you have your 4-inch pots, you get them going, they're germinated. Now you got to separate them. So let's go to the next section. And so here's another video. Hopefully this one won't be as choppy. But here I have some flowers I need to separate. And so I'm getting them out of the pot. Their root structures really well, so I just need to separate them. And so it's a little slow. And these videos, I put these videos on YouTube, so if you want to just look at them, or on your own. But essentially what I'm doing is I drop the 4-inch pot just to disturb the roots on the bottom, and I'm just slowly separating them one at a time. Again, like I mentioned, the soil is where all the plant has to interact with its first organisms. It's building its relationship, so these microbes and whatnot. And so the root is essentially where the intelligence of the plant is. And so as we're pricking them out, I'm trying to keep as much of the root structure as I can. I'm not too worried if some of it breaks, so, because especially if, can we back up and come to the, back to me. I have this tomato here I was going to do this with. So here again is a pot of tomatoes, and let's just move this out a bit. I find it super useful to tickle or to just disturb the soil as best as I can. Dropping it, I find it seems a little violent, but again, they're pretty tough and they'll manage through. But because there's so many plants that are intertwined together. Their roots are gonna, some of them are gonna rip, and that's okay. There's you can grab them by the leaf, or I just tend to grab them by the bottom of the stem. It's okay if they break. I know we all want to keep as many as we can, but sometimes they do break, and that's okay. This is why one of the reasons I enjoy this method is because I can propagate a lot of material in a set amount of space, and there's always room for things to die. And that's the only way we're going to build experience. We want to keep as many of these plants as we can alive, but it's okay if they die. Again, they're racing, not, these are the plants that essentially are using us to grow them. But they're really just all seeds are trying to survive. And this is just part of the chaos of seeds. So I have my individual plants now. Again, I've been taking them from the stem. And I feel pretty comfortable with this, so that one I kind of just unintentionally, there's still roots attached, but I ripped half the root system apart when I was trying to pull it. It still looks pretty good, but yeah. So once we have our little pots or our little plants, depending on what kind of container you want, you could use these are a 2-inch pot, which again I find very useful, but you can use a cup, you can use a jar, you can plant it directly into a bigger pot where it's going to live permanently. But I want to be able to allow the plant to be slightly above the cotyledons. In this case, the cotyledons already fell off this plant, but let me see if I can find one with cotyledons. So again, cotyledons were the seed, this first layer of this leaf here. That's the first seed that reached the surface to start photosynthesizing. And out of that, it takes a few days of just kind of, it looks dormant for a few days up on the surface, but really it's just channeling the sunshine into creating true leaves. I would not prick any of these out until I can see the true leaves, because that once the true leaves are there, then the plant has started to really come to terms with its own genetic identity. One thing I haven't mentioned and I want to mention before I forget is labeling. Label all your stuff, because you can try to remember, and even for myself, I have all these mental notes, and then a couple days goes by, and then all my mental notes just merge together, and I don't remember, was this a cherry tomato or was this a pineapple tomato or whatnot. In this case, I still have the label. Things that I include in my labels is the name of the plant, the date, and then the seed company. Those are the 3 things I consistently put on, because I find it super useful. In the last couple of years, I've transitioned to popsicle sticks. They work really well. Also, I prefer them over plastic. But the drawback is that unless you really draw or really write your material, your handwriting really dark and firm, there's a potential for it to disappear. Plastic tags seem to be fairly normal, but plastic and pollution and all that, I just transitioned over to the popsicle sticks. Yeah, and then you would repeat this over, and then you'd have a whole flat of tomatoes to give away to your friends, to plant in the garden, share, etc. Any questions on this? There might be questions. Oh, that's.
There's a question about what is a true leaf and how do you identify it?
Okay, that's a great question. Let me, where did I had those sunflowers that were in the, all right, so the true leaf, give me a moment. I'm gonna go see if I can find these sunflowers. Okay, here's two examples that I got. The cotyledons will always be the first two leaves or the first leaves that show up. So if it's a dicot, it'll have two leaves. If it's a monocot, it'll have one leaf. So, but here we have some sunflowers. And so these leaves here, I'm gonna separate. I'm gonna just clear up a little bit. So again, here's our root system. You can see these are really well developed. Sunflowers you normally wouldn't grow like this, but I did this for this workshop. So I'm going to separate these. So here we have the root system, and this is the cotyledon energy. This is all the energy that this seed needed produced to try to get to the surface. So once it gets to the surface, it needs to drop this seed coat. And this is again where the water does, it helps ease this process of softening this up. And this seed got dry again, so it's not as pliable, but eventually it would break out. So these two seeds here, or these two leaves, those are the cotyledons, right? They're just turning green because they haven't even really reached the surface. They were under that, under the seed coat, but cotyledons still nonetheless. Here we have the first set of true leaves. So you have the cotyledons who become the first solar panels. And then after a few days, you begin to see the true leaves, which they will come perpendicular to the cotyledons. And that essentially will give you the characteristics of those plants. So similar families will start off the same. So all the brassicas, radish, broccoli, kale, cabbage will all have similar cotyledons, and then you will get a distinction of what those plants are once the true leaves come across. Here's another example with this cucumber. So these are the true leaves, and again, this will let you know that it's a cucumber. But these yellowing leaves, the ones that are starting to fade out, those are the cotyledons, and so those are the ones that came out of the seed that started this whole process. And they will be different for all plants, but here's a tomato. Here's the cotyledons, these shriveled leaves, and then the true leaves of what the plant actually looks like. And that, again, when you have that set of leaves is when I would start pricking them out.
There's another question while you're talking about this about when to transfer plants from small plants into larger plants, or when to plant out from a pot into the ground.
These, for something that's a little bit stronger, like these sunflower seeds, I would just maybe start putting these in the ground. At this point, their biggest, they might fall to predation, but they're strong enough to live on their own. It's mainly something eating them. But they're ready. They're super crowded. They got true leaves. I would put these in the ground. For these tomatoes, I would give them a little bit more time to strengthen the root system. But these could also be put into the ground now. They just need care to make sure that they don't dry out. But it really is an issue of resources, so it could be time. Here's another tomato. And so here, these already, they got, in this case, they have three pairs of true leaves. That'd be more than ready. And it also comes down to your own time ability, right? When do you have time to do all of this? This can take me anywhere between 15-20 minutes just to put them all in their own cells. And again, it took me about a minute per pot to get this started, but then I'll have to spend another 20 minutes to 30 minutes putting them in their own pots. Again, do you want to invest your time at the beginning of your sowing or later on? I find this method much more easier because again, if I'm using old seed, I can use them all and then work with what actually comes up. Yeah. And these are mammoth sunflowers, so they're gonna get like the head to this was like this big, like bigger than the head. Any other question? Also, laptops near soils get dirty. All right, let's go back. Any more questions on this? Now we can go back to the. The next part on this is asexual propagation, and this is quicker production of your plant material. So this is a great way to get a larger plant quicker. I didn't create a lot of slides on this, but because I didn't know we were going to get too much into it. But things I want to mention, it's not creating a child or new genetic material, it's creating a clone. And so although this lets you build your material quicker, so you can get a larger plant quicker, you're not creating any adaptations for climate change. So for a smaller project at home, a clipping or a cutting is fine. But if we're trying to, the advantages of using seeds or sexual propagation is that we're creating the creation of new genetic material is creating the adaptations that will allow us to succeed in surviving in the long run. Clones can get sick and then they all get sick of the same material. So with asexual propagation, you need a node, which essentially what that means is you need a piece of the stem that will allow itself to create new materials, new new copies of material that it has of its growing structure. So in a node, you'll have the ability to create a stem, a leaf, or a root, and that's having one of these undetermined cells will give you the access to create copies of that plant. And then in like higher lab system, they could just take one cell and put it in a petri dish, and then that's all you need. But in our process to start cuttings at home or other asexual propagation, you can use cuttings or other divisions of some sort. And I have one example here with me that I'd like to go over. So, can we come back to me? Thank you. So here we have, this is just a simple version of how to take a cutting. And this is feverfew. This could give you a cutting, but I'm not really so much worried about the type of plant, but the process. So where is the node? So say this was lemon verbena or even an apple tree, or another an aloe of some sort. So you have your stem, right? And then each place where any of these leaves come out, at the very bottom of that, you can kind of see this baby plant or this baby leaf sticking out of that. If we were to go in between that a little, so in between there, at the very center of that is where this node is at. And that is the part that we want to have for our reproduction. So you take your leaves and then you could put this under a medium of soil or water, depending on what you got. But the cut is super important. So you want to cut right below the node. So you just want, in this case, the node was there. So I cut it right below. And the reason why that is because this longer segment, if there's no node in between, it's just a vector for disease. If it gets sick on this side, disease can travel up, get to the node, and then infect the whole plant. So you want to cut it right above the node or right below the node. Or if you were cutting from above, right above. But this little nub here, this node, is where the genetic potential to create a root is. And once you get a root, then you're good to go. Because then again, as of now, if without a root, this is just, in this case, it's just a cut flower. But there is nothing, the water will still go up, but there's not really a way to absorb nutrients and create the transition of resources enough to allow new growth. Here's another example of another asexual prop, and this would be a division. So a cutting is something that you need to create roots. So you just take the stem and you need to create roots. In a division, you already have roots and shoots. So in this is a lemon balm. So here you have the top structure as well. You have a good root system, and this is just ready to, I mean, I'm gonna just do it with my bare hands, but you could just cut it in half. So I'm cutting the roots in half. And as I get to the top, I'm just gonna use my Falcos. And so this particular lemon balm was started from seed, but so it's a genetic individual to it. The other ones that grew I started these in a broadcast sowing system too, so I just split them up. But this plant here, although now I technically have two plants, they're really just the one plant. They share the same genetic material. They're clones. But this is another form of asexual prop. You have, I was able to obtain two different plants. This can grow, this can go somewhere else. They're going to continue to grow and expand, but they're the same plant. So, asexual prop. Yeah. Any questions on this? I know that was very quick. Let me see a question. With that, I guess let's go back to the presentation. So the next section is planning on the logistics for your place. So this workshop is designed for someone doing this at home. But what does your house look like? If you're in a smaller space, what are places where you can what can you use around your house for to enhance your propagation? So one thing, not everyone's gonna have a heating pad, but a good place to start your seeds to get them to germinate is on top of your fridge. If you have access to put your plants on top of your fridge, what this does is just the machine of the fridge is producing heat and whatnot. It's stable enough, and so it'll help with the regulation of the soil temperature. Places next to a window. Again, if you don't have a lot of windows, broadcast sowing, again, in your 4-inch pot until you can figure out where to put your plants once you prick them out. again, I'm a big fan of broadcast sowing again because you can do a small amount of space and have a larger quantity of plants come out of it. Always sow more than you need. There's going to be some seeds won't germinate, some seeds will rot, some seeds will do great, and then some predator, some gopher, some snail, a roly-poly will come and eat it, and then you lost it. So having extras helps, and then you can always give some away to your neighbor, local farm, garden, leave them outside with a free sign, and you'd be surprised who takes them. And don't be discouraged if your plants die. That's just part of the learning process. Some plants are much more sensitive to being transplanted. If the weather might change, we live in a climatic area. Like it was just raining last week, so that could have some later implications on crops and leaf health and whatnot. So it's just part of the learning process. So next slide. Yeah, next slide is just my info, and then I can go switch to questions and answers directly. But this image here, hoy semillas, mañana flores, today seeds, tomorrow flowers. And again, all these seeds are a product of flowers. I just want to re-emphasize that because this is the evolutionary legacy of our plants right now. And as we adapt through climate change and the climatic uncertainty, our relationship with these seeds continues, and if humans are going to survive through climate chaos, it's going to be with our plant relatives or plant friends, and this is part of it. Just whether you're growing at a massive scale or at home, it's a direct action towards climate chaos. We're not only are we allowing ourselves to feed from whatever we grow, we're also providing a food source for pollinators, for all the other microorganisms we don't actually see, and we're allowing we're caretaking for agents of change who can sequester carbon, which is the bigger thing we all of us need to do in one way or another is support the sequestration of carbon by our plant friends. So I guess with that, we can thank you for listening to this part, and then we'll go into questions and answers.
So Xochitl, there's some questions about the last section in terms of where do you find a node? Does each node of a cutting need a leaf for photosynthesis, or can you just have the stem with a node? And should it be put into water first, or can it go right into dirt?
So this is another cut flower, but essentially when you're taking a cutting, you're just taking similar plant material. So the node isn't necessarily where this leaf is at, but the node is basically a cell at the very tip of this plant. I mean, it would almost be microscopic, but and that the node is a cell that is undefined. So it has the potential of becoming a root, a stem, or a leaf. And that when we're creating a cutting, we want that node to turn itself into a root. Because if that can and by doing that, we need it to grow, and we stimulate it by putting it in a dark space and letting the plant make the attempt to set that root out. It might not all plants are easy to make cuttings out of, and that would depend on the actual plant itself. But for a general rule, the node it's found usually at the tip of where the leaf meets, but the node itself is located right here. As you can see here, this was cut above, and there was another node here, and this is cut above it, but now this is just a hollow stem, or it's just vascular tissue, and we don't want that. We want the cut to be right up, in this case, right here, because again, this is just a straw that disease can go up. There's really no more use out of this. It's just moving carbohydrate. It's just a stem, hollow vascular tissue, and what we want is we want a new root to come out of here, and while reducing the amount of disease that can infect. Because if this rots, this has the potential to rot, and it's just this whole little piece is just going to rot and take disease all the way up to the node. Once it's cut, if it's like a succulent, you want to give it a couple hours just to like dry, like so the wound heals a little bit. But for the most other things, I would just put them in water and then put them in a medium. Again, you could use your same sowing medium that you're using for your seeds, but you wanted to keep it moist but not soggy, and you were really just hoping that the plant will set out roots on its own. It's not a guarantee. You can use you can make willow tea or buy rooting hormone, and that could increase your chances, but it's up to that individual plant at that point to take the cutting. So it's useful to take multiple cuttings, and also you could have multiple nodes in one cutting. So in this case, there's the one that I started here at the bottom, and then I could take this leaf, and if it has leaves, I would remove the leaf, because you want the plant to be thinking, okay, I need to produce new cells. My leaf just could be an animal that chopped it, but all of a sudden I'm under soil. So instead of making a leaf, it's got the stimuli response to produce something new because the leaf is gone, but because it's underground, those undefined cells are going to decide, well, we need to make something, but we don't need a leaf because we're underground. We're going to make roots so we can start eating again and talking to our plant homies. But here you have 3 nodes, one here, and then another one here. And if I wanted to, I could plant this at this level, and then I would have 3 nodes with the potential to make roots.
And would you leave the top as is, or would you take the leaves off the top?
If it was, I actually would cut the leaf in half. And if there is a flower, I'm not going to cut it because I'm still going to put this back in a vase, but I would take the flower off, because again, a flower is the reproductive organ of the organism, and it just went through a traumatic experience of like being cut up and everything. It's going to focus the rest of its energy into the flower. If you remove it, it forces the energy back into the stem, and having a partial leaf will allow you to still produce energy, in this case photosynthesis, without its full potential. So it's not sucking up all the water that a full leaf would do. You just want a basic generation of photosynthesis to keep producing sugars back into the plant.
So going back a few sections, some more general questions are about seeds and soil. There have been a number of questions about, do you have any suggested or recommended seed companies, and where can you find them? Any suggestions that you'd like to?
The 2 that I try to buy from, True Love Seeds, they're from the East Coast, and Hummingbird Farm, we did grow some seeds we grew some of our amaranth for them. But what I like about them is that their emphasis on seed keeping versus seed saving. And seed keeping is more of a reference to maintaining the stories and legacies of seeds, and not just the material like you would at a regular seed catalog. In a seed catalog, you have all this genetic material, but very little story and legacy of where these seeds come from and the importance of those communities. And that's what True Love Seeds, kind of their main platform on their seed catalog, seed company, is keeping those stories of those seeds alive. So that's one that I just recommend off the top of my head. Baker Creek Seeds, I also tend to buy stuff from them. They have a really nice selection. And I tend to buy from them mainly to experiment with, because although they have a really beautiful selection of plants to choose from, not a lot of those plants will do so well here in the Bay Area. They're just really pretty. For example, that one I can think of off the top of my head was one I grew this last year was this pink celery. It looked beautiful, but clearly was not growing as well as it would in its own region. Johnny's is another one. They have some really high quality seeds, but they're really on the pricey end. Renee's Garden is another one that I tend to get stuff from. Botanical Interest. Those tend to be the majority, and other people seed swaps.
I would also add high mowing organic seeds, adaptive seeds, and Kitazawa Seed Company, which is a local seed company that survived Japanese internment and is a really great company to support.
Yeah, definitely. There's a lot of farmers in the Bay Area that grow seed for them.
So there's a lot of questions around confusion about types of soil, and both for the beginning gardener and folks who are trying to start seeds inside. You touched a little bit on it, but if you could touch a little bit between the difference of what people should get for potting soil versus germination soil. There's a question here that says, my local garden supply has playground sand and pool sand in their soil mix section. Is there one that's better than the other? And should you avoid any of them in a seeding mix?
Playground sand might be the closest thing you can find. If you could find a quarry or something, they usually have some sort of sand in bulk that is not sea salt or sea sand, beach sand. That's the main sand that I would say to avoid in your sowing mix. Is anything from the sea or anything that might chalk up because of like cement properties or whatnot. But usually if you're buying it at a nursery, you're probably not dealing with that kind of sand. Check in with the clerk for sure. But playground sand, I believe, will work because it's salt neutral, which is what you want. Do you want the sand for the structure, not so much the chemistry? At a general potting nursery, you'll have stuff that's already pre-mixed, and again, I would say those are if you don't want to go into the trouble of creating your own soil. And even within those, the only thing I would really amend with is sand, and that's probably not even necessary. It's just more my own practices that I've just kept up with, and again, because I do a lot of broadcast. And then having to split them apart, I just find the sand gives them the structure they need, but also kind of lets it fall apart. And it has really good drainage, and the structure falls apart. It's crumbly, and that's what sand does. So for me, that's super important because I'm doing a lot of these separations, and I want it to be as smooth as possible. There you can find anything from worm castings, compost that's made. Both at a conventional or a byproduct of like the wine industry. You can have grape compost, which is wine residue. A conventional, the three big things that I would want in a sowing mix is one, structure. and that is both like drainage and water retention. And so the peat moss or coconut coir can help with that. Drainage could be perlite or sand, and then some sort of nutrients, which is compost of some sort. Again, a standard potting mix already has all those things together. The only thing that I would just caution again with the potting, just a general potting mix to use for your sowing mix, is just larger chunks of biomass. And that's like wood chips and, large biomaterial that hasn't broken down completely. You want your seed to be able to push through anything.
There's some questions about moisture and light. One question is, what is the best way to ensure a sponge-like moisture? Do you water from the top or from the bottom?
That's it. Watering from the bottom is a lot more labor intensive because you have to like move things from, like, if you have a tray, especially if you're doing this indoors, you'll have your tray and you'll have to like even or just facilitate your entire trays that you're sowing, but it'll give you a good consistency all around, versus when you just water from the top, you'll get the water percolate through, it'll drain out, and it'll dry quicker. If I was going to be gone for a day or two, I would probably soak them underwater from below, give them those 2-3 days I won't be there. But because the idea is that there's enough moisture that's the ball should be waterlogged, or if you soak them from below, and then after a few days they'll dry up again. That would be more from a convenience on my part if I wasn't going to be there. The plant themselves would probably prefer a daily attention of some sort, so watering from above would be good. And it also gives you that relationship with your plants. You get there and like, okay, I'm going to water you this time of the day. But the best way just is just to feel it. If you're, let me see. If this was my broadcast, actually use this one here. For this one, for example, how I test the moisture, I tend to use the back of my finger and I just feel it against the soil. If it feels moist or wet, cold, then I'm just like, well, there's still moisture in there. If it feels dry, or if there's like a little layer of that's kind of dried up on the surface, then I know I need to water. I also like feeling it against my face. You feel the moisture directly, especially if you, but yeah. And the visual cues from the plants, if they look droopy or wilty, then you know they're thirsty. Yeah.
So we are at 11:30. There's one more question that maybe one or two that may sort of be fun to wrap up with. One question, which is sort of an overview in maybe 2 or 3 sentences of the whole workshop, is can you go into a little bit of detail about the best environment for germination, inside or outside, covering with a plastic cover, etc.?
Again, consistent temperature and moisture are much more important for the germination of your seeds to begin with. So putting like a plastic cover on your containers helps with that, because it helps recycle the moisture in the system, and then it also keeps kind of putting a little blanket on your seeds, so that helps. For the bigger picture, it's easier to start things inside, because this is a kind of a passive system. You start them, you get things going, and then you can go outside and prep the space where they're going to end up at. And that gives you some time to work with that. But again, somewhere where you can maintain temperature and moisture will improve your chances of germination. And as far as locations, a windowsill, top of your fridge, you can start them at somewhere that's warm, your bathroom, and then move them to somewhere that's got light as they start reaching for the sun.
And one last question is, if someone wants to grow microgreens, can you answer a good way to do that?
Yeah, well, if you're going to use soil, you could, again, I would do broadcast sowing, very similar. You get all the starts. So these could actually be considered microgreens. These would be sunflower microgreens, right? And this is all it is. It's just sowing. They sprouted. They're ready, and you just come in and cut them. And if you were going to use a soilless solution, there's like glass containers, different various contractions, but a glass jar, really, you just get them started, allow some circulation of air, and then you just let them sprout, and then you just eat them. And again, when you eat sprouts, you're eating all those enzymes that are helping the plant germinate, and you're absorbing those enzymes and micronutrients into your body.
So thank you for your time today and sharing all your skills and expertise. For those who had questions that did not get answered, we are sorry we didn't have time to get to them. And if we have them, if we are able to, we will try to answer some directly to you or through a downloadable version that we'll send out to everybody about the workshop in some notes. So thank you very much. Please stay tuned for future workshops. And we are sending out a survey at the end of this work, or by the end of the weekend. Please fill it out so that we can figure out how to continue to improve this experience for everybody. Thank you, Xochitl.
Yeah, thank you, everyone. Thank you for your patience. This is my first virtual prop workshop, so I hope it went well.
Our next workshop will be a workshop called Words of Wisdom from a Farm Elder with Wendy Johnson. It will be on Sunday, June 7th. Please stay tuned for that. Thank you all. Have a wonderful rest of your weekend. Thank you, Xochitl.
Thank you.